Morning. If everyone could take a seat, we're going to get started. Last chance to get a coffee, tea before we kickstart. So good morning and welcome to our inaugural CRI's Look into the Future of Immuno-Oncology. My name is Andrew Tsai, and I'm the co-chair of the Board of Trustees for CRI. And I want to thank all of you for being here today to make this a complete IO ecosystem. In the audience, we have healthcare investors, we have the media, and we have expert panelists from both academia and industry. In my day job, I'm also an investor, and I'm really excited to be in the audience today to listen to what the world's leading experts have to say about the likely evolution of the IO landscape. So first and foremost, you know, as a human being, which we all are, we've all been touched by cancer, and certainly it's what brought me to CRI. I lost my mom to cancer when I was a kid. I've had a couple battles with cancer myself. And so this is a real personal, deep connection for me, this mission. And it's the main reason that CRI is the main focus for my philanthropic time and resources, and why I'm going to continue to do so until all cancer types have been addressed. So let me tell you a little bit more about my experience with CRI and what blew me away to join their cause 7 years ago. So CRI was started 65 years ago on a view that harnessing the immune system could be the ultimate cure for cancer. Now, for the first 55 years or so, there wasn't much proof of this. And what's amazing about this organization is that they persisted and believed well ahead of the evidence. So in the audience, how many investors here today can take a 50-year view? A 50-year bet. It's amazing. Not many people can do that. So CRI has always been focused on primary research and was the pioneer, and for decades, really the only game in town for funding grants to leading scientists around the world that wanted to test their theories on immunotherapy. Even our CEO, Jill O'Donnell-Tormé, is a scientist herself. And this speaks to our complete obsession with being a scientific organization. This focus on primary research is the reason our scientific advisory board, many of which you'll listen to today, are really the who's who of the space. These aren't new relationships, but long-standing partnerships that go way back to a time when IO was really this fringe thing. Now today, IO is the thing. Additionally, CRI has made collaboration a cornerstone of their philosophy by bringing together academia and even competing pharma groups to work together. We are the UN or Switzerland, so to speak, of immunotherapy. And our clinical accelerator is a great example of bringing different parties together in the pursuit of saving lives. There have been some amazing breakthroughs as a result of these collaborations. If you look at the picture here, there's a picture of Sharon Belvin. So 12 years ago, she was diagnosed with stage 4 melanoma and tried all types of treatments unsuccessfully. And then she signed up for a clinical trial at Sloan Kettering with Judd Wolchok. And it was testing an immunotherapy that was created by Jim Allison. She had a complete response. She's been cancer-free ever since, and it's absolutely incredible at what's possible. So even though we've come from believing ahead of the evidence to actual evidence, this is just the beginning. We need more stories like Sharon to protect our families and our loved ones from all types of cancer. Today is going to be a discussion about what needs to be done to address all cancer forms using immuno-oncology. So if you look at the agenda today, Jill's going to kick off with a deeper understanding of CRI and what we are doing to continue our leadership in the IO field. Then she's going to moderate a panel with members of our scientific advisory board, including Jim Allison. He recently won a small award in Sweden, I think, for his life's work, as well as two other legends in the space, Phil Greenberg and Bob Vonderheide. We're going to have a Q&A session, so hold your questions for the Q&A. We're definitely going to get involved, and feel free to ask anything you want, and we're going to have a short coffee break. Afterwards, we're going to have an industry fireside chat, moderated by CNBC's Meg Terrell, and it's going to include George Yancopoulos of Regeneron and Awny Farajallah of BMS. Have another Q&A session where you can ask more questions. And then finally, we're going to close with an announcement of CRI's most recent STAR grant recipients. And these these folks are really the future of CRI, of immuno-oncology. And so remember their names because you'll be reading about them in the not too distant future. And then at noon, we're going to have lunch. I encourage everyone to stay to kind of mix and mingle with all these leading experts in the space. I'm also happy to talk to anyone who wants to get involved in CRI in any small or big way and kind of share my experiences on what it means to be a trustee at this great organization. And so we have a full-packed day with a great lineup of speakers. And without further ado, let me introduce Jill O'Donnell-Torme, the CEO of CRI.
Well, it's my pleasure to be here. And as we know, today's discussion is about the future look at the IO space. But I thought it would be important to set the stage first and see where we are actually today. I don't know if everyone knows about this, but CRI has developed the first in-depth quantification of the IO landscape. And this is a capability that CRI uses to guide us to use our philanthropic dollars in our clinical strategy. But it's also a resource that's available and I think valuable to the entire field. So this is just a slice of our IO landscape, which really just shows PD-1 or PD-L1 combination trials and the growth that we've seen since 2017 to 2019. There's been an 86% increase, and this is no surprise. Everyone knows that there's an incredible increase in the number of immunotherapy clinical trials, but this is a quantification of it. And obviously, there's also been a 79% increase in the targets that these combination trials are looking to combine with PD-L1. So we more recently just reported and published in May another look, and this was a slice looking at cell therapies. And in this most recent analysis, it certainly shows that from 1993 to 2019, there's been a real explosion in cell therapy clinical trials. In fact, in that period of time, there was over 1,200 initiated. And obviously, in the past decade, you really can see the growth has significantly increased year after year. And you probably can't see all the different types, but the light blue color there signifies the CAR T cells. So obviously, CAR T cell therapies are dominating the space with CD19 as the most popular target. So obviously, immunotherapy is changing the landscape. It's changing how cancer is being treated. And it's certainly true that you can see that right now there are 19 approved immunotherapies by the FDA in the 6 categories that we use in our landscape. And that's covering over 20 different cancer indications. But how did we get here? Well, I'd like to think the Cancer Research Institute had something to do with this. We were founded in 1953 by Helen Coley-Knotts, who was the daughter of Dr. William B. Coley, who is considered the father of cancer immunotherapy. CRI was obviously a pioneer, and we were always singularly focused on immunotherapy, decades before the current wave of excitement and enthusiasm. So I am very proud that CRI's long-term dedication and support of immunology and tumor immunology has played a significant role in laying the foundation for where we are today. And I now believe that certainly the entire scientific and medical community would all agree that the immune system plays a role and is able to play a role in the treatment, control, and potentially cure of cancer. But this was certainly not the case even 10 years ago. But CRI stayed the course, and we are now reaping the benefits of that persistent vision. But as we know, the majority of cancer patients are still not yet benefiting from the immunotherapy, and there's a great deal of work to be done before we realize its true potential. So CRI is going to remain committed to the mission that we've always had. We believe, obviously, that the support of basic and translational research is critical, and that this will really yield, it's from this that we get the basic discoveries that will really yield the impact for the entire field. Our unique clinical strategy is really a collaborative model that's really helping to inform and de-risk immune-based combination therapies and accelerate development. We also have ability to convene the world's leaders in the immunotherapy space, and this really helps to position us as a global thought leader. And more recently, while we still remain primarily a scientifically focused research organization, we have recently taken on the role of being a trusted third party to cancer patients and their caregivers as immunotherapies have moved into standard of care. Now, our biggest asset, I would say, for the Cancer Research Institute is our Scientific Advisory Council. And these are the people that really guide all our programmatic decisions and recommend the people and the projects that we should support. These are led by Jim Allison, who's the director of the Scientific Advisory Council, along with these 5 associate directors, all of who I'm sure are not strangers to this audience. The SAC includes 4 Nobel laureates, with Jim being the latest one, and actually 24 members of their National Academy of Science. So we continue at CRI to fund the entire spectrum of research, from the lab through the clinic. And we do this here. This just is showing our major research programs. Our Irvington postdoctoral fellowship programs is the way that we are really supporting and training the next generation of cancer immunologists. And these are the people that are doing a lot of the basic laboratory discoveries. Our clinical laboratory integration program is a translational program that's really more focused on projects that are taking these basic discoveries and applying them into developing new immunotherapies. Our clinical strategy is called the Anna Maria Kellen Clinical Investigator, and as I said, it's a unique collaborative model, which I'm going to talk about a little bit more later on. More recently, we've added 2 new programs. Our Technology Impact Award. This is the first time that CRI is supporting not biomedical research, but rather recognizing that it is advancements in technology that really underpin the ability to take major leaps forward. So these are seed grants that we're giving to cutting-edge new technologies that we hope will end up being applied and adopted by the entire field at large. And then most recently, we've started the Lloyd J. Old's STAR program. Lloyd J. Old was our longtime director of our Scientific Advisory Council before Jim, and we have named these as Scientists Taking Risks. And you'll get to introduce our first class at the end of today, and we're very excited about this as support for mid-career scientists. Let's see. So I think it's important to give a little bit more detail about what CRI's funding has happened over the last 6 decades. And we look at this over kind of decade by decade. You certainly see trends in what was supported and what has been really important as the foundational discoveries from where we are today. So from the 50s to the 60s, it was really about looking at Coley's toxins and trying to see if we could confirm what Coley had originally seen in the 1890s. So it is a lot about nonspecific immune stimulants and the hint that perhaps Coley's toxins work through an immune function. It was really in the 1970s with Lloyd Old guiding us, and he used to always tell Helen, and I knew Helen back in the day, that science really needed to catch up to her father. So it was really Lloyd who put CRI on the path of being truly dedicated to immunology. And so in the 1970s, it was really about really basic immunology, understanding the components and how the immune system really worked. And it really wasn't yet applying it to cancer. In the 80s, I think a lot of the focus was around T cells. I think we came to understand how important they were, what they see, how they see. Antigen processing and presentation was a great deal of the kind of our portfolio of what we were supporting. In the 90s, we finally started moving a little bit away from basic immunology and doing a little bit more cancer immunology, but never leaving the basics, and really trying to understand what are the targets, what are the potential cancer-specific antigens. So antigen identification was a big part of the portfolio then. And it was only in the 2000s when I really felt our Scientific Advisory Council felt there was enough understanding about the immune system and how it really applied to the cancer question that we could really start supporting more clinically relevant cancer immunology. And a lot of this was focused on developing therapeutic cancer vaccines, but at the same time recognizing how important the immunosuppression that takes place at the tumor site is and how can we overcome that. And now in this decade, I think it's all come together. It was during this time that we've actually seen the first clinical successes of active immunotherapies, and our long-held belief that the immune system could be used as a way to treat cancer was really validated. So CRI did get it right, but continues to support the research that gets us to the next level. So one of the things we're doing, and I wanted to talk about this, is our clinical accelerator. And this is a collaborative model that serves as an incubator to study promising immunotherapy combination trials. And we work with a network of about 90 clinician scientists coupled with our IO landscape. And together, this helps us to define the most pressing clinical questions and identify and prioritize innovative research-driven clinical trials of novel combinations that can address these questions. We then negotiate access to the drugs that we think need to be brought together to be combined for biotech and pharma. We get access to their pipelines, and we also negotiate agreements with them on financial returns so that any drugs tested in the clinical accelerator, if they reach certain milestones in successful commercial advancement, we get some returns, and this comes back to our venture philanthropy model. We then partner with other not-for-profits to really be able to carry out and execute these multi-center clinical trials. And all of the trials actually have a good deal of funding for deep correlative analysis of what is happening in the patient so that we can learn as much as we can from every one of these patients. Now, since 2013, we have invested over $110 million into this clinical accelerator model. That has supported 15 trials, 2 of them platform studies, and I think moving forward we're seeing platform studies are the way to be most efficient and effective of using our philanthropic dollars to have impact. Over 600 patients have been treated in 23 different cancer types, and we've obviously partnered with a variety of companies and not-for-profits. And our primary not-for-profit collaborators are the Ludwig Cancer Research, Parker Institute for Cancer Immunotherapy, and the Canadian Clinical Trials Group. And this is just a list of the current for-profit companies that we're actually in contract with right now and working with the trials that we're doing. I want to take 1 minute just to explain one trial that I think exemplifies the power of this model under kind of a not-for-profit umbrella. This is the PRINCE study that was done in collaboration with the Parker Institute, and it's a trial that was for metastatic prostate cancer, and it actually tested 4 drugs, 2 standard of care chemotherapies, along with 2 immunotherapies, one a CD40 agonist and the other an anti-PD-1 antibody, nivolumab. And I think this shows you that amazing speed that's there. From the concept, which was back in November of 2016, agreement signed, and IND submission in 6 months, which is a pretty amazing feat to accomplish. First study trial site was initiated in July. This study had 7 different sites. It was led by Dr. Bob Vonderheide, who you'll hear from later. And we were able to, just recently in March, present at AACR the initial results from the first 24 patients in the study. It's looking like it has, at those 24 patients, have gotten very tantalizing, exciting data that looks like the response rate is better than what was seen in just chemotherapy alone. Since March, we have fully accrued the phase 2 of this study with another about 70-something patients, and we will anxiously be awaiting this year the analysis of the rest of those and keeping our fingers crossed that the initial data will be carried out with this larger number. So this is exciting. It's great news for the pancreatic cancer patients, and I think it just shows you the power of our clinical accelerator model. So in preparation for this, where do we go from here? And I think prior to this meeting, we actually surveyed 100 CRI scientists. These are members of our Scientific Advisory Council, scientists that we're funding, to get their viewpoint of what is coming down the pike and what are the important things. In terms of near term, no surprise here. I think one of the major issues is how do we turn cold tumors into hot tumors? How do we optimize the checkpoint blockades that are FDA approved at this point? And how do we move cell therapies into the solid tumor space? A little further on, I think there's going to be efforts beyond just the checkpoints that we have today. There's a lot of work looking at trying to be more personalized and identifying neoantigen predictions and using those to develop maybe off-the-shelf or personalized medications. And obviously, we need to look really closely at what's happening at the tumor site, the tumor microenvironment, the stroma. Genomics and epigenomics play a big role, and obviously metabolomics and the microbiome. So all areas of active research which I think will really inform and probably lead to the next generation of immunotherapies. And of course, big leaps, I think there's still questions that the scientists felt that can artificial intelligence be used to deal with the big data that's being generated at this point? Is machine learning a possibility? Can we develop models that are non-animal-based models that may be better predictors of what's going to happen in clinical trials? And obviously, technology. But underlying it all, I think in all of these cases, we heard over and over again, but we can't forget the basics. Though we're doing all this work in the clinic, it really means we still have to come back to the lab. There's still a great deal to discover about how the immune system functions and works and interacts with cancer. So for that reason, it really fuels us, and we will be using this for CRI to really guide where we're going, and I think we'll touch on these topics in the panel discussion coming up. So I just want to close in saying how you could help us. Obviously, we're an organization that relies on philanthropic investment from individuals, foundations, and corporations. I'm always happy to get checks, so if anybody wants to support us, I'm here. You can find me anywhere. Obviously, we also have our IO landscape. It's something that I think is useful perhaps to you or your clients, and we can do customized IO landscapes, so please reach out to us. And there are other engagement opportunities. We're always looking for new trustees. We have an associate board, which is for kind of the younger generation that we're grooming to become full-time trustees, and also, obviously, corporate partners. So with that, I'd like to invite our panelists up. If you guys can come on up. We have Jim Allison, who from University of Texas MD Anderson, Phil Greenberg from Fred Hutch and the University of Washington, and Bob Vonderheide from the University of Pennsylvania.
That order. Okay. this.
So they keep us straight.
Yeah.
These plastic chairs.
Jill. For people standing, there's seats over here.
Oh, there are seats, yes. You don't have to stand.
just
There
sat
are
on my
seats, because
croissant.
we're going to be here for a while. So thanks. I think maybe we'll start off by each of you maybe just introducing yourself and telling you the areas of interest that you're. These are all members of our Scientific Advisory Council. So these are the guys that I've known for years and years and years that have really helped move CRI forward. So Jim, why don't you start?
I'm Jim Allison. I'm with Pam Schromer, run the cancer immunotherapy platform at MD Anderson. And my specialty was I developed and at CTLA-4, began the development of CTLA-4 and continue to do both mouse work and participate in some design of clinical trials to look at combinations and try to improve the efficacy and understand why they're failures and come up with a rationale for moving in a logical and science-based manner to raise the response rate.
Bill?
Yeah, I'm Phil Greenberg. I'm at the Fred Hutch and University of Washington in Seattle. Our lab has been involved in trying to manipulate T-cell responses to target tumors. A lot of this was originally done with cell manipulation and developing techniques for expanding cells outside the body and then giving them back to create effective immune responses. In the last 15 years, really, it's become more about synthetic biology and genetic engineering. So we are now engineering T cells outside the body and then giving them back to patients, trying to create immune responses that the patients can't themselves generate, but that you can now make an immune response better than a patient could otherwise generate and create effective responses for cancer.
Okay, thanks, Jill. My name is Bob Vonderheide. I'm at the University of Pennsylvania. I'm the director of the Abramson Cancer Center there. It's a relatively new job for me. I've done it for 2 years. I've been at Penn, though, for 20 years. I think I met you upon my arrival at Penn, thinking about the CRI. Many thanks to the CRI. As you described, you were in it before it was hot.
Right.
Speaking of cold to hot, you knew it was hot all along. I bring the perspective of a cancer immunologist, immunotherapist to my role as director of the Cancer Center. It's really an amazing time. We've been pleased to be at the epicenter of what's going on. I've been focusing our laboratory work on diseases where we haven't had much luck, and certainly immunotherapy hasn't garnered an FDA indication yet, and we focused down on pancreatic cancer, as you heard the clinical trial I've been involved with pancreatic cancer. Maybe we'll return to that.
Okay, great. So let's start kind of big picture. We'll start on checkpoints. Obviously, hottest area, Jim. You opened up the field, got the Nobel Prize for it. So what do you think are the biggest challenges immediately ahead of us in terms of optimizing checkpoint treatment?
Well, I think one of them is what Bob just mentioned, is getting pancreatic cancer, glioblastoma, and some of the other ones that have not responded well yet to respond. I think that the field is beginning to settle around in checkpoints, at least a core of anti-CTLA-4 plus anti-PD-1 because of their unique and not overlapping properties that make them a really great combination. And to that's gotten us to almost 60% in melanoma, 60% of durable responses lasting 5 years for the combination. Probably it's going to go out to 10 or more, given that ipilimumab by itself has 10-year plus at about 20% of melanoma. But anyway, and then we're going to have to start adding additional checkpoints. We've learned in the last few years there's a couple of dozen other molecules that have some promise there. They're not all going to be, game changers, but they can add a little bit, I believe, and but they're expressed in different kinds of tumors, we're learning. And so it's going to have to take an individualized basis of really looking, using biopsies to see what kinds of things are expressed in different kinds of tumors and on what cells, and then putting together a package that makes sense. And of course, there's adding it to radiation and chemo and things like that that are coming.
So Bob, do you think checkpoints are going to be kind of the backbone of treatment going forward, or is there places where we're not going to use checkpoints?
It's right now hard to imagine not having a checkpoint backbone because of the biology that Jim and many others have revealed. But by themselves, I think we're realizing the limitation of stacking checkpoints. I totally agree with Jim about PD-1 and CTLA-4. But as we continue to stack, I think we'll see the rate of responses not increase as much as we want. And we're thinking that one has to actually generate those T cells in the first place by a variety of mechanisms. And there's many mechanisms that have been tried over 20 years. And many of them fell out of favor. And if you think about it, they came online when we didn't actually have CTLA-4 or PD-1. So it's a little bit back to the future. Once you generate a T cell and you're asking it to destroy a tumor, the problem of T cell exhaustion, the problem of optimally priming are before us, and we have great drugs for that now, PD-1 and CTLA-4. So it's in the near future, a checkpoint backbone is for non-cellular therapy, and even maybe for cellular therapy eventually, is what we're looking at, I think.
So I know, Phil, you're more a cell therapist than a checkpoint person. So obviously, great successes with cellular therapy and hematological malignancies. I think, obviously a big thing, can they move to solid tumors? And I guess I want to hear what you think needs to happen for them to go to solid tumors, and do you need checkpoints with cell therapy?
So, I think, the advantage obviously with cell therapy is, as Bob is alluding to, is you generate the response so that rather than try to activate something that in some cases, unfortunately, is just not present, you can essentially synthetically generate the response outside the body by having selected a target that the tumor does express and give those cells back. Now, I think in the early phases of this, we are doing it in the context of adding checkpoints. But one of the real advantages in cell therapy is that you have the opportunity to genetically engineer the cell so that the checkpoint can already be excluded. And so, for example, it's very easy to knock out PD-1, and so that you don't need to then provide a reagent that has potentially some systemic toxicity in terms of its blocking PD-1 on every cell in the body. So you can engineer cells so that they now can function in an environment where they're getting those negative signals. And our lab and several other groups are now looking at ways actually to co-opt some of these negative signals. So, for example, we can take PD-1. We know that when a T cell is being active in a tumor, it stimulates, unfortunately, the tumor to upregulate the inhibitory ligand PD-L1 and drive a negative pathway into T cells. So one simple thing is we could genetically knock out that pathway. But we also can create synthetic receptors that, for example, have the PD-1 receptor on the outside, but instead of having the inhibitory signal that PD-1 delivers, we actually change the signaling pathway so that now it actually delivers a positive signal. So that now when the T cell gets into the tumor, instead of seeing PD-L1 as a negative checkpoint, it actually sees it as a positive signal. And so we can, and PD-1 is just the beginning of that. We've reported other ones already, and other groups are looking at a panel of signals. So I think synthetically we're going to be able to make T cells that really can work in the solid tumor environment. And we're actually getting ready to start a trial also in pancreatic cancer targeting that. And again, it's the first generation is just the T cell receptor. The next generation are these synthetic biology approaches.
So I guess I'm a little bit biased. When I think about solid tumors, I'm always worried about the antigen. Obviously, CD19, perfect antigen in hematological malignancies. You can attack your B cells, it doesn't matter. I don't think to date we have such an exquisitely expressed target for solid tumors. So can we get over that? I just think, these are very powerful immunotherapies, off-targeting. You have that antigen somewhere you didn't know it was, you got major problems for a patient. So what do you think on that?
So obviously, I think one of the real advantages now with the advances in molecular biology is you can look at a tumor and you can say what genes does this express and how do those differ from normal cells. And so we've been able to identify several candidate targets. I think what you're right about is that we are probably going to need to target more than a single antigen for many of these tumors. So you may need to put in T cells with 2 receptors in it, or 2 different T cells, one with each receptor in it. I think what you said is correct. I mean, turns out that CD19 in a sense is a perfect target, but it's perfect only because the fact that those T cells actually see normal B cells doesn't impact the patient's survival.
Right, exactly
so
right.
you can live without them. And the same is true with a target that's being looked at in myeloma now, BCMA. Again, it looks to be very effective, but it's because you can live without your normal plasma cells that, again, you can get away with that. For the solid tumors, there's not many tissues. Prostate and breast represent tissues where, in fact, you can essentially target a self-antigen, and because you can live without a prostate or a breast. And so if that's the price of having an effective cancer therapy, that's probably going to be considered an acceptable toxicity. In pancreatic cancer, we think we have found a target, mesothelin, that can be targeted, but there will likely be escape from that. We've already seen that. We have a study that's about to get published actually on Monday in Nature Medicine targeting WT1 in leukemia. But there is potential escape from that, so we need, and we have now looked at alternative targets that may be ultimately necessary to build on those kinds of therapies.
All right, so we've obviously talked about overcoming the suppression with checkpoints at 2 different spots with CTLA-4 and PD-1s. We're talking about priming T cells either by a vaccine maybe or by cells. But what about, when we talk about turning cold tumors into hot? That means, the T cells are not infiltrating the tumor bed. So what are the current thinking and strategies of how do you get T cells to traffic? How do you get them to infiltrate? What's the most exciting things there?
Well, one thing that we've shown that's just that CTLA-4 itself can do that in prostate cancer and in kidney cancer at least, and maybe some other cancers. For other ones that are harder, though, there are ways coming up with chemokines or even a light dusting of radiation to activate the vasculature in the tumor and allow extravasation of cells into it. And there are a lot of more specific things coming along, I think.
Anything to add?
Well, we're impressed that a properly activated T cell can find its way into some of the most hostile tumor microenvironments. And if you have a poorly activated T cell or a dysfunctional T cell, even if you take down all the barriers, some of them physical, some of them metabolic, some of them signaling mechanistic, that T cell will still have problems. And so it was one of the ideas, a hypothesis we had when we brought CD40 agonist into treating patients with pancreatic cancer, where 90% of those tumors really are void of T cells. And by activating those T cells with CD40 and some other agents and releasing some of the brakes with PD-1, clearly in the mouse, we're able to deposit huge numbers of T cells into an environment which is among the most hostile described yet for cancer. Now, can we do better? Sure. Can we get rid of the macrophages and all the other mediators? Absolutely. But if you don't have a properly activated T cell, either in vitro or in vivo, I think we're up against it pretty badly.
So T cells are critical. We know that. Now, let's talk about, you hear about the microenvironment. You have MDSCs, you've got B cells. Yeah, but what's the latest thing that in terms of do we need to target them? Do we need to get rid of them? Do we need to inhibit their suppressive activity? And I guess also what role something like an NK cell plays in relationship to a T cell? Any takers on that?
Yeah. Well, so I think we are increasingly understanding how the cells and the components in the tumor microenvironment impact a T cell response, whether it's one that we provide by cell therapy or we activate in vivo. So there are many things now that I think will change whether a cold tumor will become a hot tumor, and it's there are pathways, the beta-catenin pathway, the TGF-beta production, the CDKs now actually are also but we have reagents now that can target each of these pathways and will make these tumors essentially less hostile for an immune response. So I think we will be able to do that. And I think as Bob has shown with anti-CD40, rather than, for example, necessarily get rid of some of these cells that are in the tumor microenvironment, you can modify them so that rather than being anti an immune response, you can actually recruit them to be part of a T cell response. And we actually showed in a paper that just came out in pancreatic cancer, for example, that depleting macrophages is actually worse than, for example, trying to activate them. That you can actually recruit the macrophages to be a potent anti-tumor effector mechanism, as opposed to getting rid of them because the way they're there naturally, they're interfering.
One thing I would add to that, we're talking about a lot about pancreatic cancer, so we'll stick with it for a minute. The number one tumor microenvironment feature that is, at least in the mice, the genetically engineered mice, that's causing a cold tumor are the macrophages. And you can deplete, you can re-educate. But what we've learned, I think, over the last couple of years is that they're there in their current state, these macrophages, because they're instructed by the tumor cell itself. So when we talk about the tumor microenvironment, T stands for tumor, right? So let's not forget, and as a pancreas cell becomes mutant with KRAS, from that moment, it begins a signature, a gene expression pattern that brings in and poisons macrophages. We can alter, and often that is caused by either genetic or epigenetic reasons for the tumor cell to secrete chemokines that bring in and mold macrophages in that way. If you go up one stream and damage that pathway in the tumor cell, or prevent those chemokines or cytokines, and we've shown this now 3 different ways, the macrophages go away, and now the T cells come in, sometimes without any additional impact. And certainly you can now treat those tumors with immunotherapy very well. So the tumor cell is instructing the microenvironment, and it's probably different tumor to tumor to tumor. Back to Jim's point, where we need to have precision immuno-oncology to know what are those pathways.
Yeah, so kind of going to like biomarkers. I mean, obviously, we have all these complicated, there's a rheostat, I think, it's positive, negative, it's not all or nothing. You can modulate this and that. How are we, I mean, obviously the goal, wouldn't it be great when a person gets diagnosed with cancer, they get their genome and their tumor analyzed, their personal, their immunology, and being able to say, what does this patient need for a personalized immune-based therapy that's going to treat their cancer? This requires biomarkers. Biomarkers is kind of a buzzword, everybody hears about it. Not so easy. Biomarkers are very different from targeted therapy biomarkers. I think when the immune system, you're dealing with a system. There's multiple. So are we going to a lot of work looking for biomarkers. Are we getting anywhere? Is there one that can be used?
It depends on what you mean by biomarker. There's a sense of having some magic thing you can pick up at baseline that's going to tell you whether they respond. That's not happened and is not, in my mind, not likely to happen.
That's what okay.
And so, but a different sort of biomarker that is the signature, not only a single thing, but the signature of what's going on, the whole symphony, of the T cells and the right kind of macrophages and the whole thing coming together well. And one of the things that is really disappointing to me, you showed there were 2,000 PD-1 plus something trials going on. Almost none of those are based on any kind of rational understanding of what the interaction between the two things would be. And unfortunately, if there's not a clinical signal, nothing is learned because nobody looks for the biomarker. Nobody looks, they're not taking biopsies from the patients that maybe fail and don't give a clinical signal to learn what was lacking. I mean, we've got a pretty good idea of what the overall successful response looks like. And if you add something, and you get partially there, that's worth knowing, and you can instruct the next thing you do. But if you don't know that biomarker in a sense, then you've gained nothing.
Well, certainly I think that, from obviously I'm going to plug CRI. In our trials, we try to make sure we do these biopsies and get, and blood and really analyze what's going on. And I think there's a movement for more of that happening.
Yeah.
But I guess you need big data sets.
Yeah.
And
Well,
how do you analyze all that?
No, I don't think you do. I mean,
You don't think you need big
I don't
data
think you need big data sets. I think the sort I'm talking about is just hints of where you are. Like we've found it's in many instances looking at somewhere around a dozen to 20 samples, and you see you see signs that tell you. I mean, it's not a p-value, but it could be a signal that says, hey, this is the overall thing that you need to look at.
Yeah, I think, I mean, I'm a bit on the other side with big data sets, because I think it's really is giving us a lot of clues about who will respond and who won't respond and why, and also we're learning why that is, and you may be able to make the switch from a non-responder to a responder. I think, for example, there was just a study published, I think, yesterday in head and neck cancer, an oral squamous carcinoma, that showed that you can do, with actually machine learning and artificial intelligence, pull some of these large data sets together and actually very accurately predict what the 5-year survival will be for that cohort. I think the problem we have right now is that the biomarkers are very good at telling you the likelihood of response. So as a physician in the clinic telling a patient that this is likely to work, that's very comforting. The problem is that there's no biomarker that excludes a patient from responding yet. And so that's the real difficulty. So you can collect all this data, and does that mean that you should say, no, you won't get this therapy? That's where we're not right now. And that's where we need to get, because these therapies are unfortunately expensive and have some risks of toxicity. So we need to really, and the more data sets I think we get, the more information we collect, we will ultimately, I think, be able to learn some of that.
Well, I think down the road that's true, but now
it's not true now. Absolutely.
Yeah, you can't do it like prostate cancer. We came up with a new strategy just simply based on looking at untreated after treatment and seeing what was missing and then what's still missing, and then you can move from there.
I think we want this to be a reality, and we're far from it. But you mentioned, we've done this before, which was with targeted therapy.
Right.
And as where precision oncology got its name. And we're looking back to that experience to see how was it successful. And it involved technology, which was brand new. It involved doing those assays in a routine way, in a CLIA-certified way. The assays were performed and sent out to clinicians before we understood every last mutation. The results are examined at every major institution by a molecular tumor board, which was a feed-forward information back. This begins to look like laboratory medicine and a lot less like my postdocs on Saturday morning. And we need to discover in that setting, but when we start ramping up in a throughput, we will have high-quality data that can then be analyzed in the way Phil was talking about. So I'm optimistic that we'll get there. We're going to need different technologies. It's not just DNA sequencing, but it does include DNA sequencing and RNA sequencing and ATAC sequencing. I mean, those are some, and we know how to use those technologies, but we need multiplexed tissue analysis, circulating tumor analysis, cytokine analysis, and many other things, all of which can become CLIA certified. And I think that will avoid us from generating a bunch of bad data.
Yeah. So I'm going to switch gears here. What about the microbiome? We hear, there's been studies that show that different people with different microbiomes react differently to checkpoint blockades. What do you think? Does it? Is that real? Is it?
Oh, it's real.
It's real. It's definitely real. I mean, we've been working with Jed Wargo, who's one of the pioneers in that, and it's definitely real. Where to take it, not so sure yet. There's some simple things that we have learned, though. There's a group at MD Anderson, for example, that learned how to do fecal transplants to actually fix patients that had seemingly, colitis. They couldn't fix any other way. Patients who were just refractory to any kind of immunosuppressant deal with it, but you give them a fecal transplant in 48 hours, they're perfectly good. That's in clinical trials now. But there's some interesting work that's coming out besides the idea of identifying one or a few bacteria that would be good, and that's seeing what effect things that people are doing now are having, and there's some data that's emerging that will be published soon, I think, showing that people that take just any old probiotic off the shelf, very often don't do so well.
Right.
Because we don't know enough yet about how to predict it. So trying to do this at home without it's not a good idea
don't
right
do
now.
it at home.
Don't
Don't
do
go
it.
buy-
Don't do
don't
it
go
at
buy
home
your
without
milk
somebody
right by-
telling you.
No, I think that's a really important issue. So there are trials. I mean, there are like the bifidobacterium. There are some bacteria that look like they are important for promoting a response. I mean, and I think, as Jim alluded to, I think there's absolutely no question that the microbiome plays a role in whether you respond to an immunotherapy or not. It even actually plays a role in whether you respond to chemotherapy or not, which I think has been an enormous surprise. But the data is very clear. So the role of the microbiome in responding to cancer treatments is unequivocal. The problem is that we are in the infancy of trying to understand that. And I think the scary data is, as Jim was alluding to, is that people, based on that, have been taking probiotics with the idea that that would make them better responders. And the data actually showed it was the opposite, that the probiotics people were getting, and essentially in these off-the-shelf health food stores, was actually interfering with the response. Now that was itself quite surprising, but it is telling us that you can't do this by trial and error. This has to be essentially science-based, and it has to be a really defined question that you're asking and trying to answer.
Okay, so in terms of moving on a little bit, better models for research, discovery, drug testing. You know, I think I know what Jim's going to say. We need to do a lot of small phase 1 studies, get some signals before we go out. But you know, how useful are animal models? I mean, in terms of predicting, do we need new models? Should we go directly to phase 1 with some, you know, basic data without going through animal stuff? Or you know what?
I don't see how we can go to phase 1 without animal models. I think they're absolutely necessary and will continue to be. I mean, everybody says, well, they don't exactly give you the details of what's going to happen in humans because they're different. That's true too, but you know, they certainly can tell you the big picture. You know, the general things that you need. And if you don't have something, I mean, people say again and again, well, why can't you just do it in tissue culture? Well, you can't. I mean, the immune system is too complicated for that. You've got too many interacting components that we don't know how to do without it. And
Are there any synthetic models that come close?
Well, I think, you know, the organoid cultures now, where you can actually grow in 3 dimensions tissue from a tumor, is actually really useful. I mean, it looks like it's becoming increasingly useful now for studying the use of drugs, particularly some of the targeted drugs. It's just, the problem is trying to, and you can ask some questions about whether an immune response has the potential to be beneficial. The problem is you can't, as Jim's alluding to, you can't really recreate a whole interactive immune system in a dish. And so you'll get some insights, but you'll also lack some information that you can't get without doing an in vivo model. But the in vivo models are becoming much, much better. I mean, the genetically engineered mouse models now really do replicate a lot of the issues in human disease. And I think, as Jim suggested, they're not perfect, but increasingly we're understanding what their imperfections are, and you can build that in. So, for example, the lack of adequate numbers of mutations in these genetically driven mouse models. So now, for example, Tyler Jack's lab is adding this mutator phenotype so that they will have some of that. So I think increasingly the animal models are going to be more useful, and they're actually going to become closer and closer to what we need to know to understand and predict what will happen in a patient.
I don't think there'll ever be a single animal model that's useful.
For sure
That's what people say. What's the best? Well, depends. Depends on what the question is.
Well, Jim should tell us which animal model he used for discovering
CTLA-4?
the anti-tumor effect of CTLA-4 if we just want to get a dose of reality here, which was is a transplantable model.
Yeah. So I'm going to open it up now to questions from the audience. Any questions? No takers? Yes. Are those microphones working behind? I think if you go to that microphone there.
Thank you all of you for the great contributions to the field of immunotherapy. My question is directed to Dr. Greenberg. So we know that CAR works in the hematological malignancies very well, But we also learned that, from the certain TCR trials, beyond melanoma, it has shown some efficacies. But what exactly is that the TCR therapies and CAR therapies indifferent in terms of homing the tumor? Can you give us some insight into that?
Well, there's very substantive differences between a CAR and a TCR. So a CAR, which is this chimeric receptor that has an antibody on the outside and has a synthetic signaling module, works very well in certain settings, in particular, as we talked about in leukemias and lymphoma, B-cell leukemias and lymphomas. But it is much less sensitive than a T-cell receptor. So first of all, it can only target surface molecules. And second, it needs a lot of surface molecules to be a signal. So whereas a TCR can see essentially all these internal proteins that get processed and put on the outside, so the range of antigenic targets with a TCR are much broader, and they include much of the genes that are the proteins, intracellular proteins that are driving the malignancy. But because they see fewer molecules, their signal is actually kind of weaker. So what we're trying to figure out is how can we synthetically design cells that will be more effective. And so in solid tumors, there are CARs now that are showing some activity, certainly in the animal models, and that are being tested in people. There are TCRs that are being tried in solid tumor models that have looked increasingly effective. So we're going to get there. It's again, this is a field in evolution. When we realize how recent it is that all this data can be shown to be effective, but now that we have a signal in diseases of efficacy, building on that is really different than going back 15 years and saying we have no signal, nothing. So going from nothing to something was a huge roadblock. Going from something to something better is a whole different kind of set of experiments, and the field's going to move very quickly.
And I think that's true of all the immunotherapies, because we now know it can be done. It's just a matter of putting the right things together. The big burden is done now. I mean, that's what this celebration's all about here.
Right.
We're on the right track now.
Any other questions?
That's a question.
One more? There's one.
So my question is regarding oncolytic viruses. I think we've seen some very intriguing evidence that in combination with checkpoint inhibition, they can be effective, potentiate the activity profiles of checkpoint inhibitors, and perhaps most intriguingly, can attenuate the toxicities associated with certain checkpoint inhibition strategies. But what I wanted to know was how you think they might be optimally deployed from an administration route perspective, the IV versus the intratumoral route, and also whether you believe that oncolytic viruses per se might potentially become the principal combination regimen modality, or if you feel that there are already other modalities that have emerged in combination with checkpoint inhibition that have superseded what we have already seen so far with the oncolytic viruses, or if it's just too early to tell. Thank you.
Long question. Well, we've got in my laboratory a little bit of experience with oncolytic viruses, and I've learned a couple of things. One of them is just killing the tumor cells and releasing antigens is only one of the things you have to have happen. The other thing is you have to really get engage the innate immune system if you want to get particularly systemic immunity and make it really work. I don't think it's going to take over as a major thing in all types of cancer, but for some cancers, where metastases are readily accessible for intratumoral delivery of the viruses, it's kind of a no-brainer. I think in terms of systemic administration of them, that'd be a lot better if there's some way to tag them and focus them on going to and finding the tumor and being really, really specific, which most of them now are not.
It is a strategy we were talking about before. What do you do with a cold tumor? It is a strategy for creating inflammation in a tumor that can then potentially recruit in effective responses, particularly if you're doing something like providing a checkpoint that actually will enhance the generation or activity of those responses. So it clearly has a role. There are other parallel technologies that are being pursued. So there are nanotechnologies that, for example, you can inject intravenously, and people are learning, as Jim was alluding to, how to tag these particles or even modify the viruses so that you could give them systemically, and they would at least preferentially wind up at tumor sites. And so I think oncolytic viruses are a very promising strategy for converting a tumor, but it's not the only one. And so which of these strategies will become dominant, or whether there will be non-overlapping settings in which one is good and the others, and alternatively in a different setting where different technology is going to be good, I think we don't know that yet. But these are definitely promising strategies for changing the way a non-responsive tumor can become responsive.
Well, I think we're probably out of time, so thank you very much for
Thanks,
all your
Cheryl.
insights. And we're going to take a 5-minute break now, and then we'll be back for our industry panel. Thanks very much.
Thank you, everyone. That was such a fascinating discussion. I can imagine folks didn't want to come back in from coffee because there's so much to talk about. So we'll continue the conversation now, and I think this is such a wonderful order of events because, of course, things begin in the laboratory, and then pharma and biotech often take it up, although I'm sure George would say you originate many things in your labs as well. But you take that science and that research and you turn it into products. And so this is going to be a wonderful discussion. I'm sure you know these folks here on stage with me, but we've got Dr. Awny Farajallah. Did I get that right?
That's correct.
All right, from Bristol-Myers, the U.S. head of medical for Bristol-Myers, and George Yancopoulos from Regeneron, Chief Scientific Officer, President, and Founder. We've already been having a fascinating discussion, and we're going to open it up to questions in about 25 minutes, so please get ready and come to the microphones and join in and ask questions. Let's begin by kind of setting the stage for where industry is in immuno-oncology. And Awny, let's start with you. Where do you think we are in terms of the timeline of immuno-oncology and really reaching its promise?
So certainly, we are still at the beginning of the immuno-oncology. There are a lot of things that we will need to continue to do. We've heard of things for Jim and the panel before. We have made a lot of strides, getting patients a lot of hope for survival. I think we have a lot of more work to do. A lot of more work for patients that are developing IO resistance, a lot of work with covering other platforms and new platforms where we are able to get there and drive to this long-term survival promise that we're thinking about.
Mm-hmm. George, what do you think?
Yeah, I think it is so first of all, I think it's one ecosystem. I mean, we work so tightly with so many people in academia, and we're all part of the same effort, so I don't think there's separate efforts in academia and industry. We're all in this together. And I think it's appropriate and it's a great time to celebrate the major advances and the breaking open of the immune approaches towards cancer. But while we can pause and celebrate a bit, I think it's also critical for us to reflect a little bit on how hard the challenge really is and how hard the biology and the cancer really is here. And I think we all have to recognize, I think Bob touched on this a little bit, is in most cancers so far immunotherapy hasn't really made a dent. Despite the thousands of trials that Jill referred to, most of them have not really shown much advance over some of the early data with the checkpoint inhibitors. And the combo approaches to date have been disappointing thus far. And you look at some of the biggest cancers, prostate, pancreatic, colorectal. There's really little advance in those areas. And so we need to all together in our ecosystem up our game and figure out how to take this promise and really deliver and hopefully deliver a lot sooner, because I would actually say I am when the first data started coming out on checkpoints and so forth, I thought that by now we would be, by 2020 we would be a lot further advanced than we are now. And I think we all have to up our game and figure out how to really take these early advances and take them to the next level.
So I do have a different opinion here, though. So I think it's really important. I think we've made a lot of advances. I mean, the work that Jim has done, the work that we have done with PD-1, the combination therapies, we have seen advances in the therapeutics of lung cancer. We have seen advances in the therapeutic of melanoma, renal. While we have not cracked the codes on some of the tumors like pancreas and prostate, there is definitely, with the combination of CTLA and PD-1, we have seen overall survival in renal cancer. We have seen overall survival in melanoma. We're still exploring in different tumors. So I think there is definitely advances. No, do we need more work? I agree. We definitely need more work, and there are certainly things that we will need to continue to do, but we can't do it alone. It's going to have to be to continue to collaborate, as George was saying. But I beg to differ a little bit on where the advances that we are made.
Mm-hmm. How do you, as leaders of organizations, make decisions about where to allocate your focus and your money? We saw at the beginning that there are more than 2,000 combination trials going on. Obviously, you want to follow the science, but you also have to think about the commercial landscape, I assume. How do you decide where is the right place for you to focus? George?
Well, we go where the science takes us. We generally don't use commercial insights to make the decisions for us. It's all based on the science. And I mean, the thing that I'd like to touch on is I do think that there's a lot of new exciting approaches that weren't brought up in the first session, certainly ones that we're approaching because we thought that they were great opportunities years ago, because we all know these take years to take things forward. So we're obviously very impressed with the cell therapy approaches. I think they're very exciting, but complementing them, and in some cases maybe opportunities to improve on them, are these bispecific approaches. And so we've invested heavily in that because we thought that the science warranted that. And they often also afford great opportunities for combinations with foundational, particularly PD-1 type therapies and approaches. And so you may have seen, we recently at recent European meetings updated our latest data with our bispecific, and they're now producing data that is looking very competitive, if not more impressive than CAR T therapy. In fact, we've tried it in CAR T failures and now seen very high response rates. So we really think that new science, new approaches that can really take things to the next levels, layered on top and in combinations. We've actually shown impressive combination opportunities between these bispecifics and the PD-1 class in particular, and we think these can unlock the immune system to now start giving results not only against the liquid tumors and the hematologic malignancies, but also against solid tumors and also prostate and so forth. So we're very excited about the future, about new approaches and new technologies.
Awny, how would you describe Bristol-Myers' take on cell therapies and bispecifics? You got into cell therapies in a much bigger way with your acquisition of Celgene. How would you just describe the way the company has sort of evolved in that thinking?
I think with the acquisition of Celgene, and we're two separate companies till it actually happens, I think that gives us a frontier of to go to cell therapy, including both it was mentioned, the BCMA and the CAR T, the CD19. I think now that we will have hopefully the platform accessible to us, we'll be able to go more broader in cell therapy.
Mm-hmm. What about bispecifics?
The bispecific technology is a technology that we've been looking at. It's not a technology that BMS has worked with.
I see. And is Regeneron exclusively in bispecifics? Would you go into cell therapies, CAR T, things like that?
No, we constantly are comparing and testing. So in our own labs, we are heavily invested in CAR Ts and bispecifics and ways of we actually believe you can combine those as well. And get actually more specificity without sacrifice and more efficacy without sacrificing toxicity. So we think that you can't be in this game unless you have all the weapons at your disposal. Plus we have major collaborations with Bluebird and with Datasets in the CAR T space. So no, we're believers that it's going to take all sorts of different weapons to really try to do a good job at addressing all of these cancer challenges that we're all facing, and you have to pick and choose the right weapon. Not, I'm into bispecifics, so bispecifics are going to, it's the right hammer for every nail. No, and, cell therapies, it's the greatest and the latest. No, checkpoints by themselves or alone. No, I think it's going to be the right collection of approaches for the right setting. And having the ability to have all these weapons at your disposal and choose the right one for the right setting can allow us to start getting, I think, much more impressive responses in the right settings.
So I think it goes to the precision medicine that we were talking about earlier, is that how you're able to find the right medication for the right patient and with all the tools that are available to us. But it is not, from my perspective, it's not one company that can do this. It really takes the whole community together to be able to do this. Partnership with academic centers, partnership with our hosts like CRI, and also a partnership within the industry. If we're not able to do this collaboration together and get these pieces together, we're not going to be able to advance the science.
I thought the discussion about biomarkers that you had was really interesting, and the idea that there isn't a biomarker that excludes patients from responding. And perhaps you brought up the idea that patients who are failed by therapies aren't tested for biomarkers to figure out what might have gone wrong. I mean, how much are you guys doing that and is that important for drug companies to figure out why patients don't respond, or if there's a marker that would exclude them from responding?
I think it's critical. I think it's critical to understand the patients that are not responding. We have a lot of ongoing work right now and our big platform of area of interest is IO resistance and patients who are not responding to IO therapy. Again, we're collaborating with Jim's labs and other labs to try to understand that. But most importantly, we just opened our site in Cambridge that is focused exclusively on IO resistance, which we have about 300 researchers across different disciplines looking there. The most critical thing here is that it is not just to understand the exclusion, the resistance, but get to the patients and do exactly what Jim was telling earlier, understand the journey of the patient, the biopsy journey of the patient, what the tissue, how things are evolving. What do I mean by that? So what happens before the patient starts therapy, after they fail, and try to understand what's changed and how we're able to address it. And as an example to that, we have just announced a collaboration with one of the Boston Medical Center where we are looking prospectively at exactly what was being said earlier, is getting the patient before diagnosis, getting biopsies throughout his journey, and trying to do heavy translational medicine, trying to do the translational work that we need to understand why patients are not responding. But what's most exclusive about this as well is that you need to get to patients that are from different backgrounds, because patients that are clinical trials may not necessarily reflect the patients that are that we treat day to day, whether from ethnic standpoint or from gender standpoint. So I think that allows us, when you go to inner city and some cities institutions, to get those patients and try to understand the biology and why they are developing that resistance.
George?
Well, I mean, there's nothing new here. This is what we've all been trying to do, forever in every field of biology where you have any sort of treatment. You try to understand why patients are responding and why they're not, and if you can understand the biology and the mechanisms behind that, you can then, of course, try to turn non-responders into responders and increase your rate of benefit. So we've all been trying to do this in every field, and certainly in cancer, since we've all been studying cancer. And of course, it's challenging, and every time you have a breakthrough, it'll take the level of benefit to patients up another level. So once again, it's fundamental biology, nothing really fundamentally new, other than that we all have new tools and new capabilities and new treatments that we will now be querying this way. But this is what science has been around doing forever.
How much are you looking at the microbiome in terms of its role?
Obviously the microbiome is important and going to be proven important for a lot of different settings and indications. But I think it's sort of like everything else. Right now it's one of these hot areas, and it's oversold, and too much value is being given to it, I think, right now. We're going to certainly find areas and applications where understanding and dealing and manipulating the microbiome is going to have a benefit. But it's like any one of these fields in its infancy, that it's going to cure everything, like stem cells were going to cure everything a few years ago. And we're going to find out that right now, I'm sure that almost everything that's being talked about and so forth is heavily oversold in the microbiome area.
Tell me
that's
you
the
really
truth.
think about the microbiome.
Awny, how about for Bristol?
I think we take a different approach. We are actually building a microbiome program in all our clinical trials, try to understand what's actually happening there. It is important for us to understand it, and I think we know that there is a role that we just have to systematically and scientifically approach it.
George, that does lead me to wonder, how can you be so sure that it's oversold or overhyped?
Well, because anybody who pays attention to science, this level of science, this much enthusiasm based on almost no convincing data, the way science works is most things don't deliver on promise based on no data. So I'm not saying, we know like C. diff is a disease of the microbiomes. I'm sure there will be diseases of the microbiome. But I can tell you, in our own field, as you may know, we have an important drug for allergic diseases like asthma and atopic dermatitis. There was a big story that, oh my God, the skin microbiome of people who have severe atopic dermatitis, is a disease where more than half your skin surface is covered with these horrific poison ivy-like lesions, that their microbiome is very different. Yeah, big surprise. I would think you're literally leaking fluid into your skin. Of course, your microbiome is going to be different. And there was all these people who were basing theories that it was the microbiome that was driving the disease, and if we could fix the microbiome, then you'd benefit the disease. Well, you give them dupilumab, you actually fix their skin, and what do you think happens to the microbiome? It totally normalizes. So cause and effect is really important here. So like I said, science tells you when you have little data and you have great enthusiasm, most of the time that enthusiasm is unwarranted.
But that's exactly why you need to study it, right? So that you're able to understand it.
I'm not saying don't study it. I'm just saying it's not going to be the cure-all. That's all I'm saying.
Okay, I guess I'll leave the microbiome
Bet
here.
more on immunotherapies, guys.
Well, I think we were talking about the microbiome in terms of its impact on checkpoint response, right? So what do you think of the data that you've seen there? Maybe we're just going to have the same conversation.
We can keep going forever. There's very little convincing data. But
I think we still need to we need to understand it. We need to study it. We don't know that. That's really what we need to understand.
Yeah. Okay, so another area that some folks might call overhyped is AI and machine learning. What do you think about that, George?
Well, we are huge believers in machine learning. We don't like to call it AI because there's nothing really intelligent about the approach. It's just pattern recognition. And it depends on big data, well-structured big data. And in our field, the hardest thing to do, is, it's a lot easier when you're Google and you have, essentially buying and Googling patterns of millions and billions of people. You're creating huge amounts of well-structured data that they can then take advantage of to figure out how to capture our attention and our buying tendencies. In our field, we need to create the big data, and that's the hardest part. The machine learning is not really going to solve things if you don't have huge amounts, and in many cases you really need much larger data sets than we have available to us. As you know, our Regeneron Genetics Center, many efforts that we're doing, we're investing hundreds of millions of dollars to create the data which you then can take advantage. So machine learning is not going to solve the lack of data problem.
Right.
And in fact, you're going to, when we know this is better than anybody, if you do machine learning on poor data sets, you're going to just get fooled and over-enthusiastic about the wrong theories. So machine learning is not the limiting factor here. It's a great tool. You need to create the really valuable big data sets.
Awny, how's Bristol-Myers using machine learning and big data?
So we're using it's an early technology, and the way we're using it is more in the pathology space, where we're trying to do machine learning on pathology slides, where we're able to follow the patients and we're able to kind of identify patterns. That's the big area that we're using it in. We do need large data sets to be able to understand it's what you teach the machine to do and how to analyze. That's certainly a limitation, but that's how we learn, and we continue to forge into that.
How far are we from getting to the place where you have good data sets, enough data to actually do this kind of work?
Well, I think in certain areas you will have, data sets sooner rather than later. But, it all depends on your goals and what you're excited about. And they're not going to give you the theories. They're not going to tell you, checkpoints are the right pathway. That's really still going to come from, novel, genius human insights. That's not going to come from machine learning. Machine learning is going to recognize patterns and data sets that are then going to provide you with hypotheses and with patterns that you can then hope to take advantage of. But it's, once again, it's not going to be the be-it and the end-all. It's going to be a useful tool.
Going back to the idea of trying to extend immunotherapy's reach to cancers that haven't been served by it yet. One story out of ASCO was around tumor-infiltrating lymphocytes. A company called Iovance working on taking cells that just exist in the body. You guys could explain this so much better than I could, but really not necessarily doing anything special to them, but just finding the cells that are already working and multiplying them and giving them back. What is your thought on that approach?
I think it's a new platform that is opening to us. We need to understand how it works better. Anything that we are able to understand, collaborate, understand the biology, that's the main important thing, and partner in how we can bring this to the patients. We still need to understand that.
What do you think, George?
Yeah, I mean, it's what we heard in the previous panel. It's going to be the collection of positive and negative signals that are going to drive different components of the immune system to infiltrate and attack a cancer cell. And there's, obviously there's many pathways that we're all trying to manipulate. There's all sorts of clever new approaches, turning a negative signal, such as PD-L1, into a positive signal. I mean, those are all sorts of things that I think that we're all going to be trying to do to increase, the benefit. Now that we've made, thanks to Jim, this big insight that we can harness the immune system, we got to do collectively a better job, and it's going to involve all these different new ideas that people are talking about. And I think there'll be, incremental advances, and hopefully we'll crack prostate, then we'll crack this, or maybe there'll be a big breakthrough that allow us to more broadly do this. But this is why we all need to be, investing in the research at all levels and in the science and in the clinical work to try to advance the field.
What's in your lab right now that you're most excited about in this area?
Well, as I touched on, I think that we do believe that PD-1 is a foundational approach, and we're trying to layer on top of it. Obviously, we have our own PD-1 that we think has produced exciting results on its own, but now we're layering on it with these bispecifics, which in a sense are sort of doing and allowing us to do what cell therapies do, but in an individualized manner where you can literally dial them up and dial them down. So we have two classes of bispecifics. The first class, which literally brings and activates the T cell to the tumor, which they work on their own. Like I said, on their own, they're having very impressive efficacy. But we've now shown in animal studies and emerging human data that combined with PD-1, they work better. But now we have a whole novel class of bispecifics that I think is the most exciting thing to us, which are these things that actually bring co-stimulation. So we've all heard of co-stimulation, the second signal that can hyperactivate the T cell. So we have bispecifics that activate a co-stimulatory signal only when it attached to a tumor antigen marker. And you can take advantage of combinatorial specificity there. And I know that the cell therapies are trying to do a lot of similar things, but there would be a huge advantage in being able to dial these things up and back and give them and test them individually, individual combinations, and then the convenience of being able to give them in a controlled manner to patients. So giving co-stim bispecifics with the more traditional CD3 bispecifics, layering them on checkpoint therapies, I think these are the sort of approaches that we're excited about and we think can take approaches against individual cancers to the next level.
And as you think about layering, and then you do start to worry about toxicities, and you are talking about very targeted approaches, does that take care of some of these concerns? What are you seeing in terms of the safety?
Well, that's why we think, this is why while we are heavily big believers and invested in the cell therapy approaches, we are very excited about these bispecific approaches because they can allow us to individually test pathways and dial them up to different degrees. And often in the same patient, you can slowly give one and the other. Izzy Lowe is here now, has shown with these bispecifics that you can really control the toxicity by gently dialing them up or down as need be. And if you can do that with your different combinations, I think you could attain maximum specificity and targeted activity without or with controlling the toxicity to a great degree.
I want to give you guys a 2.5-minute warning. If you have questions, why don't you head toward the microphones, and I'll ask the same question of Awny. What's going on in your labs that's most exciting to you in this space now?
So we're really excited about what we're trying to do with CTLA, the next generation CTLA, the pro-drug for CTLA, which exactly is trying to deliver the efficacy, but avoiding the adverse events or minimizing the adverse events. I think it's an exciting technology that we're seeing it is in our laboratory right now. We're also very excited about our locally, the antigen to do antigen presentation, the NLP3, the STING mechanisms. These are things that these are molecules that are helping us really create antigen and to be able to attract T cells inside of the tumor. So these are technologies that we're very happy to be working with. They are now in the phase 1 phase, and we're waiting to see what this clinical effect are. And we're also excited on some of the technologies that we are, we will be working on in Cambridge, where we're trying to understand map the T cell morphology, anatomy around the tumor. So that's the technology that we're working on to try, as we mentioned earlier, is really try to understand the 360 view there.
Mm-hmm. Okay, I've got more questions for you, but we've got questions back here. So why don't you go ahead?
My question is with respect to antibody-drug conjugates and where you see the future of that field heading. I mean, historically, it's kind of waxed and waned. People have been, by degrees, impressed and then disappointed with the potential of ADCs as a whole. And in particular, how do you see that field developing with respect to the likelihood that the success or failure of any particular ADC therapy is based on the target versus the payload? Thank you.
Well, once again, we're invested in ADCs. We believe that, the right approach in the right setting. That said, I do think that the power of, for example, either a cell therapy or a bispecific against the same target might have more promise. I think that the ADCs would have role if your target is not as specific as you would want it and you don't want to maximize anti-tumor efficacy because of the toxicity. But like I said, we are trying them and we are advancing them and putting some in the clinic in the right settings. But I do think that if you're trying to maximize tumor kill, I think some of the other approaches with cell therapies and with bispecifics have more power in them. That's my personal view.
I think ADCs will continue to have a role in cancer therapeutics. It just depends really on the biology of the tumor that you're dealing with and the combination of things that you need to put together to attack this tumor. So it really depends on what the biology that we're dealing with. And that's the most critical thing, understanding what's happening at the tumor type.
Brad?
Question for George. You touched on this a little bit, but the CD3 bispecifics, it seems that all companies are having issues with tox right now, the cytokine release syndrome, and I think it's actually very similar to what the CAR T treatments went through in the very early days with that, and they eventually got a hold on it. So I'm wondering how you feel like where you are with that today, and whether you're concerned that would limit the clinical or commercial utility of that type of approach in the future, or if you think you'll get a handle on it.
Yeah, well, you may have heard that Roche just announced with basically in terms of CD3 bispecifics, I would say that we and Roche are sort of been neck and neck in the lead in this space. And they've created a number of different platforms. And their one platform that was looking from the efficacy perspective somewhat competitive with ours is what they called their 2-to-1 platform. And they just announced today that they had, when they were trying to dose escalate, they had some serious toxicity issues, and now they're only going to be moving forward with the lower, less effective dose levels. I think that our advantage is our platform is unlike almost every other bispecific platform, it's a totally natural platform. A lot of the bispecific is really founded on a natural mouse platform that we created that helps us generate these bispecifics. And because it's much more natural, we've actually seen that we can give them in a controllable way. I mentioned that Izzy Lowy, who's here today, who's led this program, figured out a way to sort of gently dial them up. And when you do that, you gently sort of rev up the immune system, presumably killing off tumor cells more slowly, and then you can finally ratchet up to really high doses while avoiding much of the toxicity. So right now we look like, and I hope for patients' sake, that we have managed to get to very effective levels, like I said, where we're seeing CAR T-like efficacies or even efficacy in CAR T failures without the sort of toxicities that have hindered some of the other programs. So we're really pretty excited about that. I should also say the CD28 bispecifics that I mentioned, on their own we see so far almost no toxicity. It's very related, for those who are not aficionados, who don't follow all the specific molecules, our CD20 bispecifics essentially are activating the same pathway that CTLA-4 tries to unleash. But CTLA-4 blockade will only work in the setting where CTLA-4 is actually blocking that pathway, and it's working in a relatively less specific manner. With using a targeted bispecific, we actually target activation of this CD28 pathway, which is normally being blocked here with the CTLA-4. But now we can get activation even where there's no endogenous CTLA role, plus you avoid the generalized toxicity of blocking the CTLA-4 in other places. So these are all a lot of the same pathways that we're talking about. It's that the more targeting, the more precisely that you can do it, and the more I think you can dial it up and down individually, then the greater the chance that you have to deliver more benefit with less toxicity.
Mm-hmm. We're talking a lot here. Oh, are there more questions? Oh, sorry, I missed you over there.
Hi, I'm wondering if you both could talk from a high level about autoimmune research and the extent to which advances there is informing immuno-oncology and vice versa, and how you think R&D organizations are going to think about those two fields going forward.
You know, I think autoimmune research is really a critical element to help us understand how the immune system operates, but also it's the flip coin of when you overactivate the immune system. So it is actually now that we have the patients that may develop autoimmune adverse events, it's actually a ripe understanding that R&D organizations start thinking how we understand those patients, how to study them, and also how to solve for them. And certainly, it is the check and balance between the two elements. So definitely, as we start understanding immune-mediated adverse events, we learn more about autoimmunity.
Yeah, I think it's a great and a very astute question. And essentially, almost every pathway that we're looking at for cancer to try to increase targeted immune responses against the cancer, we're querying and evaluating and creating reagents and actually potential therapeutics to almost do the flip side, as Awny said, to try to treat autoimmune disease. So we do think that they're the opposite sides of the same coin, and if you can promote them in a targeted fashion, you can actually hopefully control them in a targeted fashion and treat autoimmune disease for sure. So I think it's a great question.
Which targets are most promising that you see there? Could we see a sort of checkpoint in immune disease?
Yeah, I mean, I think Jim did some experiments years ago where he actually put some essentially checkpoints into, for example, the cells that you lose in diabetes and showed that you protect against diabetes and autoimmune loss of those cells. And so, yeah, we are following up exactly on those sorts of fundamental experiments that were done several years ago now to do exactly that now with therapeutics.
And obviously, we're doing the same thing. We're really looking at this from a therapeutic standpoint.
Mm-hmm. Well, is anybody else? At the risk of opening up a can of worms when we've got only a couple minutes left, we haven't talked about pricing of these drugs. Of course, they are very expensive drugs, the branded drugs in immuno-oncology, and as they're getting combined on top of one another, how much are you thinking about that? You guys are leading science, so you'd think about it differently, but how does that come into how you think about development in this area?
I think you price your drugs based on the value that you bring. I don't think that the issue with pricing is just drug pricing. I think it's a healthcare system pricing issue, and that's what we need to look at it collectively rather than just drug pricing. So that's going to be the critical thing, this conversation that we need to have.
Right, George?
Well, I do personally think that unfortunately, compared to actually drugs outside of the cancer space, I think that most of these cancer drugs are a little too highly priced. And I think certainly because we're talking about combinations that we're going to have to be given, that we're going to have to figure out to come up with a much more responsible way to control pricing there, because you can't be layering a $150,000 drug on top of another drug that costs that much as well. And I think that, you know, we're going to have to deal with that. And if we don't figure out how to deal with it, somebody will figure out how to deal with it, you know, for us. So I think as an industry, I think, unfortunately, a lot of the industry is being propped up by cancer drug prices and orphan drug prices to the, to the negative impact in other spaces. And I think there has to be a little bit more balance in the pricing arena.
Is the answer to that, when you're thinking about it in immuno-oncology combinations, having to wholly own all of the drugs in the regimen so that you can create a new price for the combination?
Well, I think that might be one of the approaches to try to control pricing, absolutely.
But what if you don't? What if the science leads you to say, okay, well, this other company has the best one in this class, I need to combine with that? I mean, how do you work? I mean, you can't really work together on pricing because there are laws against it, but how do you figure that out?
Well, I think that we're going to have to figure that out. I mean, I think these are the challenges. No, because I don't think, first of all, I think it's wrong. And secondly, like I said, I mean, I think society won't tolerate, you know, layering on top 2 or 3 drugs at these prices. And even if it is in cancer, and even if you're providing, you know, a big benefit, I think that we have to figure out a better way.
All right, well, we'll leave it there. Thanks, guys. Thanks, everybody.
So it's my pleasure to close out our discussions this morning, really to announce and tell you about our Lloyd J. Old STAR program. This is a new program that the Cancer Research Institute initiated really at the behest and at the advice of Jim and the other associate directors of our organization, saying that there was an unmet need for mid-career scientists. There was a lot of support for young scientists, but that mid-career was a time that really could, you know, really by supporting this, you could really change the field. So we created this program to give significant and sustained funding to what we are calling our stars, because these are the people that I think are going to bring our field forward. This is a grant that is not directed to a specific project, but rather it's to the person. So these are people that have had great track records already, and that we hope by giving them this grant that they take risks, follow paths which they, you know, may not have thought of initially, and that they are going to be the people to follow and track for the rest of the year. So here they are. I'm happy to have them. Oops, sorry. There's 5 of them. We gave 5 awards this year. 3 of them are here, and I'd ask them to come up to the stage and shake hands with Jim and get a great photo op. Come on, Jim, you can come up here. You can come up here. So here we have Yvonne Chen from the University of California. We have Amanda Lunn from Oregon Health Sciences, and Greg Sonnenberg from Weill Cornell. If you all come on up, and congratulations on getting this great award. I think go over here, maybe. Where's Greg? Here comes Greg. You'll come up here and get up on the stage next to get one good picture. And then, unfortunately, 2 of our stars couldn't make it today, but they are Alex Marson from the University of California, San Francisco, and Andrea Scheitenegger from MSKCC. So we are thrilled, and we expect great things from you. And please, these are the people you should follow. These are our future, the future of cancer immunotherapy. So thank you very much.